I'd like to thank the organizers for the invitation to present our work today. For many years, my lab has been interested in the regulation of RNP granules in the context of oocyte quality. And today I'm going to tell you about the roles of phosphorylation and protein folding in regulating RNP condensates during oogenesis. So here I'm showing a graph of oocyte quality and quantity with a woman's age on the x-axis. We are all familiar with the drastic reduction in the number of follicles that occurs after a woman is born, as shown by the black line. In addition, the red line indicates the sharp increase in the proportion of low-quality oocytes that occurs starting in the third decade. Some of the qualities of decreased quality have been identified, such as chromosome abnormalities and organelle dysfunction. And today I want to introduce our hypothesis that dysregulation of membrane-less organelles may contribute to reduced oocyte quality. To consider this hypothesis, we need to first define what is meant by membrane-less organelles. So in contrast to membrane-bound organelles shown on the left, where a membrane provides a compartment boundary by which cells can organize their contents, membrane-less organelles, which are also called biocondensates, are able to form effective compartments via a process called phase separation. This process can generate spatiotemporal control over diffusion-limited biochemical reactions. In the past decade, numerous examples of membrane-less organelles have been identified, both in the nucleus and in the cytoplasm of many cell types. Examples include the nucleolus, stress granules, and germ granules. Although phase separations are a powerful mechanism to organize cellular contents and reactions, they are very sensitive to changes in physical or chemical parameters. So depending on protein or RNA concentration, pH, or cellular energy levels, RNA and proteins can phase transition among different biophysical states. Here I'm showing the in vitro synthesized Fus protein, shown in green. This is a nuclear RNA binding protein, and it's also represented by red in the cartoon below. So proteins can reversibly undergo condensation from a diffuse state, shown on the far left, to a liquid-like granule, shown in the second panel. They can also further transition and condense into a gel-like granule. A solid-like state can also be achieved, which is not reversible and is sometimes associated with a pathological state. For example, in ALS patients, mutations promote aberrant phase transitions of the FUS protein into solid aggregates, which are associated with the loss of motor neurons. In the germline, fewer examples have been uncovered to date. However, the pre-mutation in the FMR1 gene is associated with nuclear condensates of mutant FMR poly G granules in the stromal cells of Fragile X-associated primary ovarian insufficiency patients. FXPOI patients have a high rate of infertility, which is not fully understood. These and other examples indicate the regulation of phase transitions is critical for cellular function. So what biochemical reactions might phase transitions regulate in the oocyte? Given the RNA binding protein and mRNA components of RNP granules, an obvious hypothesis for their function has been in regulating RNA metabolism. A beautiful paper recently published from the Afruzi lab used the Drosophila oocyte model system to show that a solid state of OSCAR RNPs is required for the proper localization and translation of the OSCAR mRNA. When the solid state is precluded using genetic modifications, the liquid state that results confers impaired RNA localization and translation and has severe consequences on embryonic development. This example also clarifies that disruptions of the solid state can be as deleterious as dysregulation of the liquid state. In mammalian oogenesis, we don't yet have a mechanistic understanding of phase transitions. However, a few types of transient RNP granules have been identified. In one example from Melissa Pepling, she demonstrated the existence of a Balbiani body in mouse oocytes of primordial follicles of neonate mouse ovaries. In this confocal micrograph, you can see the conserved RNA binding protein trailer hitch, which is labeled in green. It co-localizes with the human homologue of this RNA binding protein, RAP55, which is in red. So we see these bright yellow granules indicated by the arrow. The Balbiani body also contains Golgi, mitochondria, endoplasmic reticulum, other RNA binding proteins, but it's a transient structure, and in the mouse it disperses as follicles grow. A second example of a transient RNP granule, or membrane-less organelle, was identified in the Svoboda lab, and this was in GV stage oocytes from 6 to 14-week-old mice. The group identified subcortical aggregates which they called SCAs, which are indicated in this middle row with the white arrows. So these were condensates that assembled in this region called the subcortical RNP domain of GV stage mouse oocytes. And you can see this is transient, as they're not present prior to the SN phase of the GV stage, nor are they present in meiosis II oocytes. The granules contain several RNA binding proteins, as well as untranslated maternal mRNAs. And this led to the hypothesis that the subcortical RNP domain is a maternal mRNA storage domain prior to meiosis II. It's not yet clear if the subcortical aggregates have a role in promoting oocyte quality, and no one has yet asked if the subcortical aggregate RNA granules are maintained with age. An example of a membraneless organelle has been more recently characterized in human oocytes as well. So here we're seeing regulated phase transitions of two PB-like proteins, PB-like 1 and 4. PB-like proteins are endoribonucleases that bind pi RNAs. They also regulate the stability of microRNAs. On the top row, we can see PB-like 1 condenses into a large granule in premeiotic oocytes. It then decondenses during pachytene and recondenses in oocytes of primordial follicles. On the bottom row, we see PB-like 4 condenses into multiple small granules at the nuclear envelope that may coincide with the Balbiani body, also known as intermitochondrial cement. This study was done examining fetal gonads, so we don't yet know if the PB-like 1 granules change with age, and we don't know if they're associated with oocyte quality. So I hope I have convinced you that investigating the regulation of RNP granules during oogenesis is worthwhile. We've seen that RNP granules are highly regulated throughout Drosophila, mouse, and human oogenesis, and we see the function of RNA binding proteins can change as their biophysical state changes, as I showed you the example of dysregulation of the Oscar RNP when it changes from a solid to liquid phase. Ultimately, my lab would like to understand if the regulation of phase transitions of RNA and RNA binding proteins and the regulation of the formation of RNP granules contributes to oocyte quality. As a first step towards this goal, my lab has been using the model system C. elegans to study how phase transitions of RNA binding proteins are regulated during oogenesis. The C. elegans model offers many of the usual advantages of small model systems in terms of small size, large brood size, and rapid life cycle. In addition, the worms exist as self-fertilizing hermaphrodites and males, and they are optically clear. In this micrograph of a living adult hermaphrodite, I've labeled the oocytes in one of the two gonad arms. Lastly, many genetic techniques are straightforward in the worm, such as CRISPR, as well as making fluorescently tagged reporter strains, which are important for live imaging of RNA binding proteins in oocytes. In the top cartoon of this schematic, I'm showing you C. elegans oogenesis in a young hermaphrodite. So you can see in the top left, there are two U-shaped gonad arms, one at the anterior, one at the posterior, and they join at a central uterus where the embryos will be found. So in the bottom of this cartoon, we're zooming in on one of the two gonad arms, and you can see labeled by mitosis is at the distal end of this gonad arm a stem cell-like population of immature germ cells. These transition into meiosis cell cycle, and they are in pachytene for the majority of this distal part of the gonad. They move around the bend, progress to diakinesis of prophase 1 prior to meiotic maturation, ovulation, and fertilization by the sperm that were made in an earlier developmental stage. After fertilization, the embryos will be found again in the uterus, which is shown here at the bottom left, and they'll develop to about the 50 cell stage before they're laid by the worm. It's important to note that in a young hermaphrodite, meiotic maturation and fertilization are occurring every 23 minutes. So this is a very efficient assembly line-like system. In contrast, on the right, I wanted to point out, before I tell you about a contrast, some comparisons and contrast with meiotic arrest in different species. So for comparison, in all species, primary arrest occurs at prophase 1. So I just showed you that for C. elegans, and that is true for insects, vertebrates, including mammals. What's different is when the point of fertilization occurs relative to the cell cycle. So the blue arrows are showing different points of fertilization. For C. elegans, fertilization occurs prior to completion of both meiotic divisions, while in most vertebrates, we can see on the right that fertilization is occurring at metaphase 2. So the important point here is that because in all species the primary arrest, indicated by those red vertical lines, is occurring at prophase 1, this commonality is demonstrating the applicability of a simple model system such as the worm for our understanding of oogenesis. So the diakinesis prophase 1 oocytes of young hermaphrodites contain both condensed and decondensed RNA binding proteins. On the left, you can see the default state of some RNA binding proteins, or RBPs, is condensed into small granules, which are germ granules. Proteins in these granules include GLH1, homologs of VASA or DDX4, and 2 worm-specific proteins, PIGL1 and MEG3. On the right, you can see the default state of other RBPs is mostly diffuse throughout the cytosol, and proteins that have this localization pattern include CGH1, homolog of DDX6 or RCK, CAR1, which is a homolog of LSM14 or RAP55, and MEX3, which has homologs called MEX3. What makes C. elegans such a useful system to study oogenesis is the extended meiotic arrest that occurs in middle-aged hermaphrodites. So I told you earlier that active meiosis involves the ovulation and meiotic maturation of these oocytes every 23 minutes. In contrast, on the right, I'm showing you a middle-aged hermaphrodite, which has used up all of its sperm. And therefore, the signal for ovulation and meiotic maturation is no longer there. And in this condition, the oocytes continue around the bend and end up stacking up. You can see they're a more rectangular shape, and they are in an extended prophase 1 arrest. This arrest can occur for several days, which is a significant percent of the worm's lifespan. We can also use a FOG2 mutant, which lacks sperm, and so these are essentially females, and the oocytes of these female worms are meiotically arrested throughout their lifespan. The extended meiotic arrest of either the middle-aged hermaphrodite or a FOG2 female can be reversed if the worm is mated by a male. Therefore, we have a very convenient system to control meiotic arrest in the lab. Important to note, in these arrested oocytes, phase transitions occur. So we observe we observe large-scale condensation of RNA-binding proteins into large RNP granules during this extended meiotic arrest. So both condensed RNA-binding proteins and diffuse RNA-binding proteins both undergo this large-scale condensation, forming these RNP granules I'm showing you on the top right. Some of the diverse RNA-binding proteins include germ granule proteins, P-body proteins, including 2 of the ones I showed you that undergo condensation in mouse oogenesis, DDX6 and trailer hitch, KH domain proteins, and untranslated messenger RNAs. And I'll show you what some of these look like in this micrograph on the bottom left. You can see that these large RNP granules in the arrested oocytes, shown in the bottom row, are up to 20 times larger than the small granules indicated by the white arrows in the top row. So in the top row, we're looking at germ granules in a diakinesis oocyte of a young worm, and they are very small. Right? They are enriched for POS1 RNA and PIGL1 protein, but relative to these much larger RNP granules of arrested oocytes, we can really see that scale and difference. Now, based on their composition, we hypothesize that the large RNP granules maintain the quality of oocytes during extended meiotic arrest. And more specifically, we propose the regulation of RNA metabolism may be changed in arrested oocytes. For example, RNA stability may need to be increased during this extended arrest. Not shown on this slide is the reversible nature of the RNP granules. If sperm is replenished and meiosis resumes, the RNA-binding proteins decondense rapidly. So to better understand these large RNP granules in arrested oocytes, we've recently used several functional assays to characterize 4 different RNA-binding proteins, and our FRAP analyses, which are shown here, indicate that PIGL1 and CGH1 have very liquid-like proteins, right, with very fast or rapid recovery rates after photobleaching. In contrast, MEX3, which is shown in blue here, has a slightly slower dynamics and MEG3 appears to be more gel-like with very slow or no recovery at all, and that's shown in red here. In a second assay, we asked to what extent the RNA-binding proteins are sensitive to elevated temperature, as sensitivity is often associated with liquid-like phases. So we compared worms at essentially room temperature, or 23 degrees Celsius, to worms upshifted to 34 degrees Celsius, and we found that only Piggle-1 undergoes dramatic decondensation at high temperatures. CGH1 undergoes some modest decondensation, but MEX3 actually undergoes some modest condensation, having an opposite effect. So these data, the FRAP data and the elevated temperature exposure, in combination with hexanediol and other stress assays I don't have time to show you today, are all consistent with a model that we have put together here on this slide, where in our model, MEG3 is the least dynamic of RNA binding protein of these four with gel-like properties. MEX3 also has some gel-like properties, but in contrast, CGH1 is more of an intermediate phase with a mix of liquid and gel-like properties, and Piggle-1, shown in light blue, is very dynamic in a liquid phase within RNP granules of arrested oocytes. So these descriptive studies have led us to ask about the regulation of RNA binding protein condensation. So we asked if either of the gel-like proteins play a scaffolding role to promote condensation of the liquid-like RBPs, but they were not required. We did uncover a role for the translational repressor PUF5 in promoting the condensation of MEX3 and MEG3. However, this role may be indirect, and it may be associated with the regulation of oogenesis. So now I want to turn to a bigger picture question about the regulation of RBP condensation. So as we consider these two developmental states, extended meiotic arrest and non-arrested diakinesis oocytes, it seems possible that large-scale condensation of RBPs could be activated in arrested oocytes, or it could be that the large-scale condensation needs to be inhibited to ensure the quality of these diakinesis oocytes that are actively undergoing meiotic maturation. So in either case, what might be some candidate regulators? Well, dozens of headlines from the literature have pointed us in the direction of considering post-translational modifications in regulating RBP condensation during oogenesis. We can see headlines with linking glycnacylation with liquid-liquid phase separation, phosphorylation in terms of regulating liquid-liquid phase separation and aggregation, ubiquitination, and there have been many lovely reviews that really summarize all of these post-translational modifications that seem to be very important regulators of phase separation and RNP granule dynamics. So in what way can these post-translational modifications regulate phase transitions? In the top cartoon, you can see that these PTMs can enhance or disrupt weak multivalent interactions between RBPs. And in the bottom cartoon, you can see a heterogeneous solution of RNA and protein in which the PTMs are inducing changes to multivalent interactions that can either exclude RNA from a condensate or actually recruit it to a condensate. Well, there are many examples studying the role of PTMs on condensation of RBPs in vitro and in cell culture. There are fewer examples in germ cells. From the limited studies that have been published, we can see phosphorylation of RBPs sometimes suppresses condensate assembly, as such seen for MSY2 in the mouse and ORB in Drosophila, and at other times phosphorylation is promoting the condensed state, as seen for GRTH in the mouse chromatoid body and GLH1 in the C. elegans embryos. So the first of two regulators of phase transitions I want to describe today is the extracellular signal regulated kinase, or ERK. In diakinesis oocytes, which are shown here in the top row, diphosphorylated or activated ERK is detected in these most proximal oocytes. And we can see this is localized strongly to the nucleus of these two oocytes in panel B and also throughout the cytoplasm. As I've shown you previously, MEX3 is diffused throughout the cytosol in these diakinesis oocytes. In the bottom row, in contrast, we see arrested oocytes, where activated ERK is not detected and MEX3 is condensed into large RNP granules. Thus, ERK activation is inversely correlated with MEX3 condensation, which leads to our hypothesis that activated ERK inhibits the condensation of RNA binding proteins into RNP granules. If our hypothesis is correct, depletion of ERK by RNA interference should result in ectopic condensation of MEX3. So on the left, I'm first showing you some control confocal slices of GFP-tagged reporter strains. MEX3 is in the top row and CAR1 is in the bottom. And in these control oocytes, we can see both proteins are mostly diffuse throughout the cytosol. In contrast, after depletion of ERK, which is the MPK1 gene in C. elegans, we can see ectopic MEX3 condensates that are very prominent in the top row. And we also see CAR1 condenses into small granules, shown by the white arrow, and then also some larger condensates indicated by the yellow arrow that resemble endoplasmic reticulum. Quantitation of these particles allows us to conclude that there are significantly higher amounts of granules or condensates after ERK depletion. We observed a similar trend when examining effects of ERK depletion on the translational repressor PUF5 and the stress granule protein GTBP1. In both cases, there were significant increases in the number of small granules that were detected. However, the degree of condensation was not as dramatic as for MEX3 and CAR1. Taken together, our depletion experiments demonstrate ERK has a role in inhibiting ectopic condensation of diverse RBPs in diakinesis oocytes. The questions we're currently addressing include genetic approaches to investigate the extent to which the canonical RAS-ERK pathway is involved in regulating RBP condensation during oogenesis. And here I'm just showing some of these upstream components in the pathway, such as MEK, RAF, and RAS. We're also working on testing multiple mechanistic models by which ERK modulates condensation. There are many possibilities, but the first model would be that we're considering is direct phosphorylation of RBPs by ERK. So as I mentioned earlier, phosphorylation can regulate the formation and properties of condensates by either enhancing or disrupting multivalent interactions between RBPs. So it's possible that ERK is phosphorylating MEX3 or CAR1 directly. Second possibility is that ERK is phosphorylating an intermediate, such as the CCT chaperonin, which is then ultimately having this inhibition of ectopic granules in oocytes. And this is the second regulator I'm going to be describing in just a few slides. A third possibility that we're exploring is that ERK is phosphorylating various substrates that are modulators of cellular remodeling. In particular, we have noted many correlations between RNP phase transitions and the endoplasmic reticulum and microtubules being remodeled. And I don't have time to go through all those data today, but they are pretty intriguing correlations that we are pursuing. And then lastly, it's possible that ERK is acting through CCT and cellular remodeling of the ER or microtubules to have its inhibitory effect on condensation of RBPs. So why do we speculate ERK might act via the CCT chaperonin? Well, several years ago, we leveraged the FOG2 mutant to do a genetic screen to identify genes that promote condensation of MEX3 in arrested oocytes. So in this cartoon, you can see the steps on the left are not particularly important, but what we're looking for is a change in the condensation of the MEX3 protein in arrested oocytes. So in the control and in most worms, what we would see is on the top right cartoon where we would see MEX3 granules, but in many positive hits, what we observed was decondensation of MEX3. So this screen was very effective, and we identified multiple chaperone families from our screen, and chaperone families have been identified in 2 additional worm screens, as well as in yeast and mammalian cells, as all regulators of RNP granules. The chaperone family I'm going to talk about today is the CCT or tri-C chaperonin family. So although the chaperonin has been implicated in all of these diverse studies, we noticed some intriguing differences. In some studies, CCT appeared to promote condensation, while as in others, it was a very potent inhibitor of condensation. So we are curious to know if it might have a role inhibiting condensation in diakinesis oocytes. And we also were curious if there was any subunit specificity because this chaperonin has 8 subunits, which I'll show you on the next slide. Okay. So there are 8 asymmetrical subunits that form 2 rings to form this sort of cylinder folding machine, which is shown in this cartoon. You can see a different view of these 8 subunits labeled 1 through 8. In this middle cartoon, we see the purple protein is the substrate. It's going to specifically interact with different subunits of the CCT chaperonin, and then in an ATP-dependent manner, the proteins are folded and then released. And then in mammalian studies, the subunits have slightly different names, which I'm showing on the right. But the subunits are highly conserved in all eukaryotes. This chaperonin is estimated to fold approximately 10% of the proteome, and its obligate substrates are actin and tubulin subunits. So does CCT regulate MEX3 in diakinesis oocytes? So we first asked about MEX3 due to our potent result with ERK, and after control RNAi conditions on the left, you can see MEX3 is diffuse, as we expect, but after depletion of CCT2, we can see these large condensates form in many of the oocytes. And these were significantly increased when we did particle analysis. We have tested 7 of the 8 CCT subunits, and we find all of them except CCT4 inhibit the condensation of MEX3. Therefore, there appears to be a small degree of subunit specificity in regulating MEX3. We next asked if CCT regulates other RBPs in diakinesis oocytes. We observed large condensates of the CAR1 protein in oocytes after depletion of CCT2, as shown by the arrow here. Of 4 subunits we've tested so far, CCT2, 6, 7, and 8 inhibit the condensation of CAR1. So it will be interesting to see if other subunits have a similar phenotype. But in any case, the penetrant and dramatic condensation phenotypes indicate that CCT modulates the condensation of multiple RBPs in these diakinesis oocytes. We next asked if CCT modulates P granule or germ granule proteins in diakinesis oocytes. And these are, again, proteins that are already normally condensed into small granules, as shown by the control RNAi picture on the left. After depletion of CCT2 on the right, we can see the GLH protein is somewhat less condensed into those granules, and when we quantitate by particle analysis, we do see a significant reduction. And several CCT subunits seem to have this effect, but not all of them. And interestingly, we saw no effect on PIGL1 condensation so far. And this is interesting because PIGL1 is regulated by CCT in early embryos. Taken together, our data indicate CCT has RNA binding protein specific functions and either inhibiting or promoting condensation of RBPs into granules during oogenesis. We are currently considering 2 mechanistic models for how CCT might regulate RBP phase transitions. MEX3 and CAR1 could be substrates of CCT directly, and their proper folding may prevent ectopic condensation of MEX3 and CAR1 in oocytes. And this would be similar to the idea of aggregates forming of unfolded proteins that can result in disease. Alternatively, the effects of CCT depletion could be indirect. An intermediate substrate of CCT, such as tubulin or actin, may be required to prevent the ectopic condensation of MEX3 and CAR1. And certainly it seems likely that an intermediate of some kind is involved in terms of promoting the condensation of GLH1 into granules and oocytes. Interestingly, from the literature, we know that the disruption of microtubules often can result in the increased size of RNP granules, and this is seen in diverse model systems, yeast, zebrafish, and fruit flies. In addition to testing these models, we are also in the process of determining the subcellular localization of CCT subunits using CRISPR to make tagged lines. We're also asking if CCT regulates stress-induced condensation of RBPs, and eventually, we would like to know if any subunits of CCT are phosphorylated by ERK. So to wrap up, today I hope I've convinced you that understanding phase transitions during oogenesis is important and understudied to date. While the C. elegans system offers many advantages, more studies of mammalian oogenesis are needed as well. Our results indicate ERK and phosphorylation are important regulators to prevent ectopic condensation of RBPs in diakinesis oocytes. In addition, the CCT chaperonin and protein folding seem to have a similar role, and sequence analyses suggest multiple CCT subunits have ERK docking domains and phosphoreceptor sites. Therefore, ERK and CCT may act in the same pathway to modulate phase transitions. We are also proposing that in old-aged or myotically arrested oocytes, the requirements for regulation of RNA metabolism may change and necessitate the large-scale condensation of maternal mRNAs and RNA-binding proteins. Ultimately, our studies of regulation are going to provide a foundation to determine if the biophysical state of RNPs in oocytes is critical for the regulation of RNA metabolism and or oocyte quality. I'd like to acknowledge all of the students who have done this great work. Mohammed Al-Aswad, a very talented PhD student, Mingzi Gao and Brooklyn Watkins, our master's students, and a very wonderful crew of undergraduates have significantly contributed to the work I've shared today. I want to acknowledge our collaborators at MD Anderson, Swathi Arora, and Deba Das, and the NIH for funding, the CGC for strains, and the funding sources and support from Central Michigan University. Thank you.